Thank you.
Well, thank you very much, and it's good to talk on home ground in Glasgow. I chose the title tonight, Plants, Petroleum and Primitive Life. It's got quite a big area of study, and so the title was chosen deliberately. We're going to talk about the plants here because one of the most fundamental geological and biological materials is the chemical which coats the pollen and spore grains of plants. And this material has been protecting modern and fossil pollen and spores for, as we'll see as we go through the talk, millions and thousands of millions of years. So it's quite an important material. It's very important in the petroleum industry, as we can see with that simplified cross-section of petroleum exploration. And down in the left-hand corner, we can see an ancient Precambrian material. That's actually from the apex chert in Australia, which is just short of about 3,500 million years old. As well as having the title Plants, Petroleum and Primitive Life, I could have called the story tonight the Sporopollenin story, but you'd have been even more mystified with the word Sporopollenin, so I thought the original title was more appropriate. Because Sporopollenin, which is the coating material around spores and pollen, hence the combined word, is the most resistant organic material of biological origin found in geological samples. So it's very, very important. And we can see down the side there, those are some fossil materials, which if you remember your first or second year geological courses at university, you'll remember the idea behind it. So pollen and spores. Pollen to most people means hay fever and sneezes. But that's only the annoying part of it. All plant materials need to produce pollen and spores to continue the reproduction and to continue the evolution stage through geological history. And this is just a sketch of the amount of pollen. You remember what a pollen grain is? It's probably 20, 50 microns in size, just enough to get into the pores of your nose or your throat to give you hay fever and sneezes. Well, one spruce tree in 50 years can produce 20,000 grams of pollen, and the other ones down there produce varying amounts. And in southern Scandinavia, each year, the spruce trees of southern Scandinavia produce 75,000 tons of pollen per year. Quite a lot. And when we think, and we'll argue as we go through, that the pollen coating, which is so important in protecting the kind of genetic material inside the pollen grain or the spore for floating around or being blown around for fertilization and reproduction of the plant, it's so important. So we have a lot of it, and it's everywhere. And as well as trees, we know from our day-to-day experience it's present in plants, but we'll see it's also present in algae, in fungi, and in lower plants as well. So this kind of special or unique material, although I'm a little biased on those definitions, takes part in much of earth science, in much life science. And these are the areas you can see. All these parts Sporopollenin has got a large to a smaller function in what goes on. We'll be talking tonight in a little bit of the plant physiology, some palynology, some geology, particularly in the geochemistry, and a little bit towards the end in cosmochemistry and what we can find. As well as the kind of probably research ideas which we just mentioned, it has wide applications, sporopollenins, in petroleum exploration, in Precambrian geology and geochemistry, and also we're finding, or we have been finding for a few years now, there are some reasonable links, scientific links, not biological links, in extraterrestrial organic matter, which we find in meteorites and we find in outer space. And we'll just touch on these as we go through. So when I started this work quite a few years ago now, we were able to look at ancient rocks up to 3,500 million years old. And at that time, we were able to push back the evidence for life represented by the fossils in these rocks, and we related it to the scientific and, in a way, the kind of theological understanding of the origins of life. And we'll try and focus towards the end and see what we know and what we think we know. Thanks, Mel. Right. So, I put this in for, having spoken just before, I put this in for kind of engineering geologists. It's to do with geological bits and pieces which you may not do in your courses. So there are many, many fossil plant materials. If you were to go up the West End into the fossil grove, you'll find these fossil trees. You can't miss them. These fossil trees are, they're about 300 or 350 million years old. They were found in, I think, around the 1880, when they were renovating the area to make the park up in the Botanical Gardens area, and they were just kind of tidying up a quarry to landscape it, etc. And they came across these very, very important fossil tree stumps. These are probably more what people think of fossils. These are real, or they can be leaf imprints on a rock. These are fern materials, probably from the Carboniferous. Here we have, this is a fossil spore here. I think it's lower Permian, about 350 or so. And this is a beastie which we struggled for quite a lot of years whether these dinoflagellate cysts from the kind of, well, from dinoflagellates, but similar to kind of algae, but they aren't algae, whether they did contain this unique sporopollen material. And in the end, we found that they did. So we added that again to our list of sporopollenin containing fossil material. I think all Scottish geological students must have been taken to or shown the Rhynie Church, which is a very famous and rich organic containing rocks up in Aberdeenshire. These are about 300 million years old. These, the centre and the right one there, these are samples from our North Sea, from our Jurassic. You can see here we have materials which are spores, which are woody materials, and which are plant fabrics, which is there. Those, believe it or not, are what we call structured materials in the samples, whereas the one on the right on the top line, this is amorphous material. So often, all the fossil material which we find in rocks and in the North Sea, as we see here, has become amorphous. It's been changed and messed up, and that's how we have to find it. But geochemistry does allow both those to be looked at, mainly the centre one by microscopy, and the one on the right there, this can be done by chemical methods as well. And then we make many, many millions of years jump. Here we had about 170 million, 160 million in North Sea. We're now jumping to rocks that are 3,500 million years old. And these, as we'll talk about a little bit later on, these are from the Swaziland Formation in southern Africa. This here is a thin section, and the black blobs and the scattered black material on the one on the left-hand side, this is organic material, more or less amorphous. But if we treat the rock carefully and well, and we have a bit of good fortune, we can find, and the bottom line on the right-hand side here, we can see preserved microorganisms. And these, to repeat, these are microorganisms, probably algal material or cyanobacteria, which were laid down 3,500 million years ago. So let's just quickly have a look at modern pollen and spores. I think pollen and spores are quite beautiful. I don't know why Damien Hirst hasn't actually caught onto these and kind of pickled them in formaldehyde or anything like that. So that they're quite beautiful. They are functional, but they're different in size, they're different in shape, and they're different in morphology. And as we will know, each of these is unique to a particular plant species. But I just want to take you through one or two pictures now of Lilium Henryii, a beautiful flower, and also a beautiful pollen grain. And on the right, on the top line, is Mucor mucedo, which is bread mold fungus. And these materials have had a big part to play in the study of what is this sporopollenin, what is its nature, what is it made of. So we did a lot of, excuse me, we did a lot of work on Lilium Henryii. It was quite an artistic research project at that time, just growing many, many beautiful flowers, watching them develop, measuring the buds and that. And we followed the development of the anthers in the plant and what was happening inside the anthers in the plant as the precursors to the pollen grain were building up and finally laying down, bottom left hand here, the Lilium Henryii pollen grain. These are the stages where the development hit in the microspores or the tetrads, and then this is where all the kind of chemistry and that's going on here in the anthers in the plant. These are carriers which from the tapetum cells, they kind of carry the precursors of the sporopollenin across in the anther and deposit it down on the anther to give this particular pattern. And in Lilium Henryii, compared with any other plant material, higher plant or lower plant, has this unique morphology. And it's due to here to the biochemistry which is taking place on the surface here, which allows these precursor materials to come down and lay down in a different pattern. In a different pollen grain, in a different plant, the actual process will be the same, but actually the deposit here to give the different shape and the different morphology will be quite different. So that's briefly Lilium Henryii. Mucor, mucedo, or bread mold. We found that the hyphae of the fungi wanted to come together On the top left-hand side, you can see that there's a thickening, and there are asexual spores formed there, and they tend to be formed of cellulose, which is quite tough in geological history, but tends to get destroyed relatively quickly. It only lasts a few million years, when this other material can last thousands of millions of years. It's all relative. Whereas when we have the hyphae coming together, growing together, we can see a thickening occurs, and a zygote spore forms here, as described there. And this material, the sexual spores, the hyphae coming together, this is formed of this Sporopollenin material. So as well as following this, we can see on the left-hand side where we have a petri dish with our plus strain of Mucor on one side and our negative minus strain on the other, and you can see them gradually going together. If you look very carefully on the left-hand side dish, you can see a band which is occurring around and towards the centre. And on the right-hand side here, you can see that it's come together, and the dark material on the right side, that is where the zygospores are formed. And what after our original biochemistry, we were confident enough and fortunate enough to be able to do some labelled carbon-14 work, where we fed into the material at different stages C14 mevalonate and C14 acetates, and we incorporated this into the chemicals that were taking part through the process, and then we took out some of the chemicals and fed them back in. And to cut probably a year's research down to one slide presentation, we found that the material which was being enhanced and produced and was required for the production of the zygospores in bread mould was carotenoids. A very well-known group of chemicals, but there's always been a big question in science that there were vast amounts of carotenoids produced annually in the oceans. It's estimated, maybe a little conservatively, at about 10 to the power 18 tons of carotenoids produced on planet Earth every year. It's a lot of material. So when we published this work, it wasn't just the nature and structure of Sporopollenin, but it was a new function for carotenoids in nature. And as well as looking at tiny zygospores or nice-looking pollen grains, this also explained another big query that was around at the time. You may have come across red tides. Throughout the oceans of the world, particularly some areas off the coast of South America and in the Caribbean and in Australia particularly, at certain times of year, the ocean, the water, changes to almost blood red, or often with a yellowy thing. That's the amount of what's happening. And we were able to relate this with some colleagues down in London, and when the algae in these oceans, when their environment, temperature or salinity changed, the natural instinct for the algae was to protect themselves, to protect the genetic material in the algae so it could survive for another day. And algae are quite sophisticated low plants, but they aren't sophisticated enough to just make the amount of carotenoid required to coat its own outside. It just kind of spews it out and uses the little bits that's wanted, and all the other stuff just flows around into the water, and that's where the famous red tides come from. We do get red tides, I think, at the English Channel, I think once or twice a year, particular temperature zones and things like this. So as well as finding out the nature and the structure, we're able to find out a new function for carotenoids. This may look a little bit complicated. It is. These just mean long chain carbon-hydrogen compound ester chains. But if you imagine that we have, this is the structure of sporopollen that we worked out, the molecular structure. If you imagine going from top to bottom of this structure are the frame of a ladder. And if you imagine going from right to left, or even from left to right, if those were the kind of the connecting bits in the ladder, and then and this would be a three-dimensional one, so it'll be ladder structure up and down, and also coming out towards the audience would be part of it. And we believe that why this material is so resistant to temperature, pressure, even to most microorganisms, is because of its structure. You can break some of these bonds, break a lot of the bonds, and there is still many bonds there holding it together. And biochemically, if you put in hydroxyl groups and such, then they tend to be a little bit resistant to microbiological degradation. So that, briefly, is what the molecular structure was. You can imagine, I'm sure you can imagine, that there is lots and lots of bits in this chemical structure that stick out, and anything that sticks out in chemical structure can change. It can change your position, it can change its stereochemistry, etc. So, one of the important things that this turns up in the petroleum industry is something which is, has a structure X at, say, ambient temperature. By the time it's been heated up to 50, 70, 80, 90, and that's the temperature where petroleum is generated in the ground, some of these will be lost. Some of these will fly off. So if we're able to follow the chemistry of that structure at different temperature regimes, we can follow and find the optimum temperature where petroleum may or was formed in the ground. So it's sporopollenin, present in spores and pollen, is used routinely now for determining thermal maturity at which sediments have been at. If you're a hard rock geologist, you want to be thinking in 5, 6 degrees centigrade plus, this won't help you, they've disappeared. If you're a petroleum explorationist, you want to be able to look at those temperatures which were around, let's say, the 100 degrees C plus or minus 10 or 20. And these are the things that get used, because the minerals present in the rocks don't change, or change very little at those kind of temperatures. So using the organic material through that. Also, we've been able to try and relate we've moved on from the palynology into the paleobotany, and down the left-hand side of this diagram on the left of the screen, we can see the age of various geological sediments, and we can see at different times and at different rates the pollen and spores developed different shapes and different morphology developed. And these are invaluable, not just to the petroleum industry, but to much of the geological research, where they can say, this is from the Tertiary or this is from the Jurassic. And you can be reasonably certain that the main determinant in there was the pollen and the spores which had been preserved as fossils in those materials. And we went on to find that the outside, on the right-hand side here, the one with the arrow, the exines, the outer wall of pollen and spores, mainly consists of this sporopollenin wall, the exine, with an inner wall made of cellulose, the intine. And we were able to see that from the bottom of this little table on the right here, the megaspores, the ratio of sporopollenin and cellulose is very high, more than 9. Sometimes it goes way towards the 20s. And then the third type of materials we looked at, there's a reduction. Then as we get into the gymnosperms, down again, the angiosperms is almost an equal amount of cellulose and sporopollenin. So we are able to work with the paleobotanists, and we've been able to put on mathematical pictures of the development of pollen and spores throughout the geological column. I haven't put it in here, but did some work at one time with a guy, and we published on the chemistry of fossils, where you can cluster and group particular species, which you see on the top there, their occurrence in the geological column, and their actual composition. And although it doesn't fit exactly, the clusters are very, very convincing. So again, I'd like to suggest to you the coming together of the different disciplines in science, the geology, including the paleobotany, the palynology, the biochemistry, the chemistry and such, is essential and very important for this kind of work. A new discipline that started to catch on about 30 years ago, in very tentative times then, mainly because of instrumentation, but now is a routine science, is organic geochemistry. Simply organic materials occurring in geological sediments. And this can cover many, many different areas. I'm going to talk a little bit about the origin and distribution of fossil fuels, a little bit about the origins of life, and Precambrian. But it's very important in oceanography. Environmental studies is often the flavour of the day now if you want to get a grant, and recent day. So you can see the discipline of organic geochemistry is vast and important. So how does this fit into the oil industry, into petroleum geochemistry? Well, all these areas we can see on this slide here are essential to understanding and finding petroleum under the ground. Surface prospecting, what has actually seeped up through the sediments to look at. Petroleum just doesn't appear by turning on the tap. Petroleum, as we'll see, is formed from organic material that is accumulated in what we call a source rock, a source of hydrocarbons. And depending on the geological or geochemical history of this source rock, the maturity, as we call it, and the generation of liquid or gaseous hydrocarbons, then we do find the migration and accumulation. When the material gets into the reservoir, we do find that the oil, or gas, oil particularly, becomes altered because of the geological conditions. And all those are brought together, not every one of them every time, but a selected choice of those materials there can give us really an understanding and a picture of our oil field. Before petroleum geochemistry became fashionable and routine, let's say 30 years, a little bit before we got the instrumentation understanding, when someone went in to spend 1 million, 10 million pounds on drilling an oil well in a frontier area, the chances of finding an oil well, leave aside the commercial, but finding an oil well, was probably about 1 in 10 or 1 in 12. When organic geochemistry now applying these kind of study methods, it's now down to about 1 in 4 or 1 in 5. And if you're in a good basin, then it can be improved on that. So that's been cut down to a third of what it was. And when you think an exploration well can be costing anything from £2–5 million upwards to drill, you can see how favourable petroleum geochemistry analysis and interpretation has become with oil companies. Next slide, please. This is just a reminder to say that this is where our source rock is down here. And here we have, below this, the maturity has been correct for liquid or gaseous hydrocarbons to be made. Above this, the source rock has not been mature enough. It hasn't been in the pressure cooker long enough. And then it migrates away, and hopefully the geophysicists and structural geologists can tell us whether there are some traps there, and we hope that the migrating hydrocarbons have been not met, but stopped by a particular fault structure or an anticline or things like that. So let's go back a little moment to look at organic petrography. It's not unlike geological petrography, but it's on organic materials, this, rather than on the minerals and such present in. If we take a representative sample here of mineral, an average figure in any mineral you'll pick up in a basin or anywhere around is there's about 3% of it can be organic material. Some of the material you can see going down from the end there, the organic material, down into the bluish part there. Some of it is soluble organic material. Soluble meaning it can flow, but we mainly mean in organic petrography, it's something you can extract out with a solvent. It's potentially, but only very small amounts, petroleum-like. Most of it is what we call kerogen. This is insoluble material which has survived in the sediment. This consists mainly, here we're going down to the yellow area on the bottom right of the picture, liptonite materials. This is formed from those vast amounts of spores and pollen and cuticle and algae that have been deposited many, many years before. And this is oil and gas prone, if everything goes right through its history and its geochemical story. Then we have vitrinite material, which is basically derived from wood. Sometime in its early history, it was probably occurring in forests and that, or the forests that have been reworked throughout the deposition of it. And this is prone to produce gas. If you remember from your school books or even your university books, you'll find that the southern North Sea is gas prone, and the source of that was an extension of the Midlands and South Yorkshire coalfields out under the North Sea and deeper burial. Whereas the northern North Sea, or just the central and northern North Sea, which is only, 40, 50 miles away, the character of the kerogen is very different, and that produces oil, and if it gets too deeply buried, it gets cracked down to produce gas. So you can see how essential and important it is that we understand what organic material, what this 3% organic material consists of. It isn't enough to know it's just kerogen, because it may produce nothing. It's important to know, can it produce oil and gas? Can it produce separately gas under the correct conditions. This looks complicated, and it used to be complicated, but I'll just briefly explain. If you are gardeners, you may know that there are all sorts of different chemicals inside the plant. Not just sporopollen coating the pollen, but all the way through in the leaves, in the stems, in the roots. And two important groups, and they're quite large groups these, are sterols, derived from steroid materials, and hopanes, derived from hopane materials. And this is a straight chain isophenolic material. And I just want you to imagine now, this is the top left-hand corner, that there are long chains represented by those rooftops, and sticking out are some lines. And they represent carbon compounds sticking out. And we can see on these other ones here, all the rest of them, there are bits that are sticking out. Some have got dotted lines on them. And that is to represent a three-dimensional picture of this particular compound. All of these compounds don't always occur in your samples, but there's always a significant group of sterols or a significant group of hopanes. And why are these sticky out bits important? For a number of reasons. But one main reason is that when something like number 6, you can see on there, becomes past a certain temperature in the sediment, the dotted line function will stereochemically change position. Let's say, for example, when it's at 40 degrees C in the ground, that function there may be sticking out of the screen. Whereas if it goes up to 65, generally the structure remains the same, but that flips over and it can be kind of pointing into the screen. And similarly with all these kind of flicker around at different temperatures and different conditions. So biomarkers, we're certain that these have come from biological materials. Well, you can't expect Joe in the lab to be making this, can you? It's complicated. So these are key things, and we can, using gas chromatography mass spectrometry, we can identify these, and not just identify it, but we can separate and identify whether that function there is sticking out towards you or whether it's sticking away into the screen. So these are key biomarkers now which we can use in all sorts of geochemistry, particularly thermal studies in the ground. Down the left-hand side, we have the stages of maturation, changing principally through heat of the sediment and its contained organic matter as it gets deeper buried. Down the right-hand side of the thing is the hydrocarbon products which we can expect to get from these materials at these maturity stages. And along the top, we've represented, remember, we had our oil-prone material, we had our gas-prone material, and we had mixtures in the middle. And we can see, as our material, look at the left-hand kind of balloon, the peak oil generation. And we can see as it becomes deeper and deeper buried, when it comes to the mature zone, this is where the peak oil generation occurs. And if we can find those places, find good migration paths, find some good structures, we made. If it's gone too deeply buried, it's gone through the oil floor and it's gone down towards peak wet gas. That's very short chain hydrocarbons, and it can go down generally to gas. Well, if we had a different starter material, like in the center, our yellow balloons, then those are the composition, our starting material there, and our maturity pathway gives peak wet gas condensate. And if we're here with our coaly material, then we get very, very little gas and lots of oil generation. So again, to bring these pictures together, starting material along the top, maturity increasing down the left-hand side, and the type of hydrocarbons we would expect to find in our basin. Note that starting material is again organic geochemistry, palynology, and things like that. What kind of spore is it? How old is it? Etc. etc. Going down the left-hand side, we need to know what is the temperature of this basin been. We saw in the last but one slide about the biomarkers, which you can use, but still determining the maturity of any basin is done by studying the spores and the pollen present in the basin. We'll miss this one now. So let's see an example of North Sea. This is from the Jurassic. The lines at the top of the black lines at the top of the burial diagram are different aged Jurassics. Those in the top half of the picture are those that have not been buried too deeply, and they've never achieved any maturation. And then as we move down there, remember these are the same aged Jurassic, in this case Kimmeridge Clay Jurassic sediments, and we can see, if we take the bottom line here, we can see that this material started to generate its oil in the mid-Cretaceous. The one at the top here, the one at the very top of the picture, that's never ever generated any hydrocarbons. Whereas as we move up the burial history, we can see that some of the material here has not generated until the Paleocene or the Eocene. So by knowing the composition, the burial history, the maturation and the generation, we can begin to map a basin and know, A, whether anything has been generated, and if so, where. But above all, we need to know when this was generated. Take a somewhat extreme example, although there are cases like this before. If we had here this lower part of the basin beginning to generate its oil, peak oil here at the end of the Cretaceous probably into Paleocene. But all the structures that wants to collect this oil in our basin weren't laid down until the Eocene or the Miocene. Well, although we've got plenty of oil generated, we've had nothing formed at that time to collect or receive the oil. You can see how the kind of picture is more complicated than a computer program sometimes is, balancing all these things at the right time and trying to understand them. Just briefly then, to say about using our biological markers. H1, 2, 3 and S1, the H1 to 3, these are ratios of 3 separate hull planes. And imagine H1 as having that thing sticking out towards you, and gradually as it becomes more mature, it wants to, the triangle here, it wants to turn and face into the blackboard. And H2 is another ratio, and H3, and this is a spheroidal ratio. And we can follow what this ratio is here, from 40 to 0, 25 to 60, 25 through all these stages, etc. And we can relate this in the oil or in the source rock hydrocarbons to temperature down the left-hand side and the maturity of the sediment. So I hope you can see how the big picture of e.g. a North Sea basin or a Gulf of Mexico basin can begin to understand it by tiny, tiny, tiny molecular components present in the sediment and present in the oil samples. Exactly. Will you go past the right. So let's move back a lot of millions of years now and look at some Precambrian work that have been involved in over the years. I was surprised, is it all right to say to the geological, I was trained originally as a chemist, but coming later on, and I had a lot of great friends who had a lot of patience with me and taught me geology as I went along. I was surprised to see how many and what areas of Precambrian deposits, Cratons they are. I thought there'd be little bits here and little bits there. But throughout the current map, there are vast areas which contain Precambrian. Precambrian, we're talking here from about approximately 2,000 million years up to towards 4,000 million years. The ones highlighted on here are samples that I've worked on, and there's now, I think, just short of 50, I think it's either 48 or 49 areas on planet Earth that have now got Precambrian rocks with organic material in them, and 15 of these 48 or 49 have got organic material in them which is older than 3,200 million years. So Precambrian geochemistry and Precambrian geology has really come alive in the last maybe 10 or 15 years, and it's still living very, very healthily. So what are these materials like when we see them? Well, this is quite a young one, 850, 900 million years, the Bitter Springs of Australia, the Amadeus Basin. We're interested in this because it wasn't just a simple, isolated microorganism which was there, although they are present, but there was clusters of cells there, and in thin sections we can see the cell walls. And when I tell you that these around here are about 15 to 20 microns in size, those the kind of microorganisms on the top right-hand side of the picture, then these materials, both their morphology, even the dimple which occurs here, and you can see it, they occur throughout the Precambrian. So we do think that probably the evolution taking place during the Precambrian, although it was very significant, particularly the cyanobacteria, has probably been, it plateaued, and much of the organic material in the sediments that we've been found has been consistent, with some variations. One of the first materials that we looked at was the Gunflint Formation, about 2,000 million years old, in North America. And there's a lot of clusters of cellular Precambrian algae there. So we've gone back to 2000 now. Good evidence of organic material. As well as the organic material present in our Precambrian rocks, there's a whole science, and a very valuable science, of stromatolites. And these occur vastly. I think of those 48, I think half of them are probably Precambrian Stromatolites. And as well, they're quite beautiful. These were derived from algae deposits and algae reactions which have caused this. This is a particular large at Yellowstone supergroup in Canada. If you see the wee guy in the bottom left-hand corner on the left-hand side picture, let's say 6 feet. Well, these stromatolite sections are up to 60 or slightly more feet high. And in some places in South Province in Canada, they stretch for 150, 200 miles. So the deposition of these, 2,500 million years ago, must have been vast geological occurrences. These were the first Precambrian I looked at, and I've always had a soft spot for the Swaziland system. Here we are on the map at Barberton. To the right of Barberton is Swaziland. To the north of Barberton is the Kruger National Park. And these rocks here, from the Zwartkoppe down to, they sit on a granite rock and trace through it. They're scattered over, here we're looking at best part of 100, 120 miles. So they're not just a little group of rocks that someone has spent their whole lifetime looking at. They're there. The picture on the left-hand side is in the Kamati River Basin. The top area there are the Moody, which are about 2,500 million old. The middle section in that photograph are the Fig Tree, 2,500, 3,100. And in the bottom of the basin, the Kamati River Basin, is the Ondervaart group. And these have been dated up to 3,500 million years old. These are some scanning electron microscope pictures and some thin sections here. These are from the organic material from the Kamati section. You can see they're definitely organic, we showed, and also they've got particular size and shape. Note the lower picture on the left-hand side there. That microorganism is between about 15 and 18 microns in size. If you look very closely, not too closely, you can see a dimple. And these are the kind of materials I was saying, they've been present in the bitter spring of age 800, 900 million years ago, and now we're looking at 3,200, 3,600 million years ago. And there seems to be very little development. Just a commercial here, we're fortunate when we publish this that picture got on in front of Nature. It was quite an interesting article at the moment. And this one we're looking here, this is at the very base of the Swaziland system, the Trace Druid Formation, which is probably this kind of age here, 3,650 million years old. How did we know they were organic? Well, we did quite a lot of chemistry and geochemistry on them. And these are taking the samples and biodegrading them and also chemically degrading them, oxidizing them, and looking what kind of components they are. It's very interesting. These are monocarboxylic acid degradation products, and these are dicarboxylic acids. And suffice to say is, if you have something which has been made abiogenically, non-biologically, then you have materials clustered around here. Somehow, abiogenic experiments don't seem to get going past about C3, C4, a lot of those. But when you're finding that you've got these products with C16, C18, C22s in this thread here, the only explanation anyone can give so far is that it must have been derived from a biological source. And then quickly moving on to a similar type of thing. This is a very popular area now in the Pilbara Craton in Western Australia. Again, it's mainly stromatolite formations in these hills here. I don't know whether you find Australia new amusing or not, but this is one of the hottest places in Australia, and it's called North Pole. But so next one. Another area, we saw this at the beginning, this apexture. We can see that these filamentous materials are pretty common in these rocks of this age. Then we come to the oldest rocks we know in the world, and these at the moment, well, those that have got some kind of sedimentary relationship. There are some igneous rocks which are pushing, 4,000 million, but things that have got some kind of sedimentary probable history. And this is up in Greenland at the Isua Supercrustal group. And these are the objects we see under the microscope. We're not too sure whether they're organisms or not. Personally, I think they are, but there are questions whether they are or not. This is an infrared, a detailed infrared spectra of some of these organisms here, and we can see the functional groups, the carbon, hydrogen, carbon, oxygens, and etc., are very pronounced and very common towards having a biological origin. So, we'll try some more organic geochemistry on them. You will all know, of course, from your previous studies, if not from your day-to-day work, that when plants take up carbon dioxide in the process of photosynthesis, during the photosynthetic process, there's a fractionation takes place, and that the carbon-12 and carbon-13 are the carbon-13 is preferentially used in the photosynthetic pathway. So if photosynthesis, e.g. biological process has taken place, then we find a delta 12-13 change. So what we did here was, this is the envelope series here on the left-hand side. The C's which we see in the top half of that picture underneath the fig tree are what we looked at for the carbonate present in the rocks. Carbonate is very unlikely to have had any fractionation due to biological activity in this area. And then we looked at the organic material in these samples here, and we can see the O's on the picture there, on the green and the top of the yellow of that picture. There has been significant fractionation from 0, and the fractionation has gone up to -30, which is very, very characteristic of fossil biological material. We had a few problems down in the area, the base here, the trace fluid. That's the bottom hit there of the yellow one. We found again the carbonate, there'd been no fractionation. And then we found that the organic matter present there was intermediate, -15-ish. And this is where things come awkward. If it had been 30, fine. If it had been nothing, fine. But when you get something intermediate, it gets a bit of a nuisance. But I worked on with a guy from Australia on this, and he looked at a worldwide suite of Precambrian rocks with the organic matter. And he found that the higher the thermal maturity that had happened, that the fractionation had switched back. There was, here we have here where it is -30. This area had been pretty hot, up to about 200 degrees C. And that caused very likely organic material which had been at -30 to retract into this -15 level. So we're pretty sure that the whole of the Swaziland sequence is biological in origin. Whereas when we go to the Greenland area here, we can see here lots of C's representing the carbonate, and we get a whole scatter of the stable carbon isotope values for the organic matter. Again, this isn't very helpful, whether it's a heat problem, whether it's not. But the scatter here is probably too large to be applying, relatively simple explanations for it. So the Isua sediment, the oldest currently known sediments of some biological origin, is still an open question. When a biological material reproduces, e.g. an algae, it forms and reproduces, and always you get the size of the spore that's formed, the constant. You don't get one that's 1 micron and one that's 100 microns. Biological processes are really clever and consistent. So what we did here is, the red line, if you can see it on the left-hand side of the diagram, it's a bit there, but those are two modern algae, Tractospora and another one, check out read, sorry. And we plotted those, and that's the size distribution of these modern algae as they developed along that. And we looked at the Komati, that's the Swaziland system, middle of the Swaziland system, and we found that the distribution of the fossil microorganisms in those sediments fit, shown by the yellow here, exactly along the line there of these modern algae. Again, not 101 conclusive, but very strong data to show that those Precambrian fossil microorganisms probably had the same or similar evolution to what modern algae have. And when we look at some other things, like here, we can see that the scatter is so wide that it's difficult to make. Here we're looking at the North Pole samples from Greenland, from Australia, and there's quite a big scatter there, which puts a small question mark, I think only a small question mark, against are they all biological or some probably inorganic. And so that's another technique which, added together, can show difference there. Now then, quickly, carbonaceous chondrites. I was fortunate enough when we were doing this work, people were, believe it or not, keen to give us meteorites, and we didn't have to go to eBay for them. They were from other places. But leading up to our work, this is about 25 or 30 microns in size. Prior to our scanning electron microscope studies of this, those look a little bit like some fossil organisms. They're a bit blurred, and if you don't look too closely, they're a bit round. They are organic. This material there is organic. But when we looked at these, we found that they had a consistent shape, but their shape did not resemble whatsoever any organic material. What they were was, if you look here, these are mineral grains, and you can see some of the mineral there. And the organic material, let's say for the moment, wherever it had come from, in the meteorites, had coated the mineral grain to give a structure. And the consistency of these, I think it's 6-sided things, based on the mineral crystal, was very, very strong indeed. Again, we looked at the top picture is some chemistry degradation products of the Henryii meteorite, and this is a biological fossil some Tasmanite samples of about 200 million years old. And you can see, again, we have this spread of degradation products up into the higher numbers, up through the teens, carbon numbers, up into the 20s, similar to these. These are not identical, but they're very, very similar indeed, which is suggesting, I'll say this carefully, I want to say it twice, that the material we find in carbonaceous meteorites is chemically very similar to the chemical composition we find in Precambrian and ancient rocks. Please note, I've not said biological anywhere in that. If I believed it, I might not say it either, but we're not sure at all. It's probably stretching things too much. But chemically, the character of what we find in meteorites and in fossils is very, very similar. Exactly. And then finally, the biggest jump tonight, or so far, and that is what's happening out in space. Well, lots of excuse me. Lots and lots of chemicals are being found by scanning the heavens. Much of this has been very small organic compounds, probably 4 carbons long, or organic compound ions, CH3 plus and that they're scanned for. And there's literally now hundreds of different small organic compounds being found in outer space. Good. But we were interested in, the trouble with small compounds is it can be contamination from somewhere somehow. The same in ancient rocks as well as meteorites as well as outer space. And also, like most things, the bigger the molecule, then you're likely to get more information from it. We know there's hydrogen out in space. If all we look for was hydrogen, well, we'd find it every day or every night. But here, we were interested in what about the large molecular compounds that we were beginning to find in space. And this is a spectra, the infrared spectra here, of an object in the Orion Nebula. Each of these dots in the lower left represent a reading. Good. Down the right-hand side of those infrared pictures, the top 3, the A, B, and C picture, those are different types of Precambrian matter and fossil materials. And the D one here is a computerized combination of all these other readings for the infrared spectra that we have. And can I suggest that the left-hand picture from the Orion Nebula, large organic matter, and the combined infrared picture from Precambrian material for the large molecular compounds that we've been looking at is not unlike each other. We know what these are. They're long-chain hydrocarbons with some unsaturation and some oxygen functions here, and we think the same kind of thing is there as well. So I'll leave it with you that we may be getting large molecular weight carbon-based polymers from outer space as well that's out there. Okay? We'll go past it this note. So the last two, bringing something together is, I hope I've been able to take you along the way to convince you that we have evidence for life way back to about 3,600 million years ago, and probably a little bit older than that. Let's draw a line there. Well, if we look at the history of planet Earth, when it was forming, 4.5 million years ago, for the first half a billion years, it was uninhabitable. And it wasn't until around 3.8, 4.0 billion years ago, that it began to settle down for the possibility of life to develop or arrive. These areas here, up to 4,000 million years ago, were so hot, we're told 600 or greater than 600, that not just that life couldn't have existed, but even the precursors for life, any amino acids, any formaldehydes, any other building blocks. So the early ideas of chemical evolution, and granted they were in the 1920s, Professor Opperan and Haldane, where they wanted to have these primeval soups, and they had almost an infinite length of time for them to react together and this to happen and the other. Granted, it could have happened the first time, but it was unlikely to happen the first time. So that time span, which the early chemical evolutionists wanted, isn't there now. We've pushed the evidence for life back to here, about 38, 37, when the Earth was in such a state, particularly thermally, that it could support precursors and life. Next one. Right. So what do we know? I think we know that on the top right here that one idea is that our original atmosphere was ammonia, methane, instead of what it is today. We know that Charles Darwin's evolution, origins of species, is proven or almost proven, with a few little gaps. Although Darwin himself said when he published 152 years ago now, he said that he inferred that he'd done the easy bit, explaining cellular and evolution. He said he didn't want ever to start and look how life originally started. And so the origins of life is still there to precede the Darwinian evolution. So what are we going out of space and what's happening? So where we are now, the science has expanded and reduced at the same time. We have the big science, where we're looking at astrobiology and cosmochemistry and physics and all that. And we have the micro, where they're looking at cellular and subcellular levels. And these things are coming together very encouragingly and very interestingly. What I wanted to leave you with was the three words at the bottom. This work I've been talking about generally, is kind of Nobel Prize work. And there's lots and lots of hypotheses comes forward. Science is mainly a hypothetical subject, I think. You're right until the next thing turns up, and you kind of go along the journey together. And that's where much of this is, and great. But when you pick up your newspaper, and if you can find it, the science report, it's usually about an inch tall in the bottom right-hand corner. The science writer in the Daily Mail or something like that will have said, life found on Mars, so-and-so, so-and-so. And what has been a very encouraging and true scientific hypothesis, suddenly, at the stroke of a pen, becomes a theory. And then, the popular science programs on television, which we get lots and lots of them, they're pretty to look at, but sometimes a little bit short on facts. That suddenly becomes a fact. And what John and Mary in the street is hearing on television is, oh, it's proven. It's proven. It isn't. Because at the movement of a pen, cross out hypothesis, cross out theory, and tick fact. We all know all the answers. So if you're interested in this, and I think most of us are, where we came from and when, read Nature or read something like that, because it's generally working in this area here, which is healthy and forward moving. Right, thanks.
Thank you. Anyone have any questions, or is there the option online for anyone to have any questions? I've got a question. With using the ratios of using the ratios of carbon 13, carbon 12 to
it's not carbon 12, it's 13 and 14.
13 and 14, sorry.
Yeah.
Can you identify when photosynthesis started? Has that been pushed back?
Yes, not simply from that, but a lot of it's been done. And one of my scientific heroes was Calvin. He got the Nobel Prize for photosynthesis, and then he went on to Precambrian stuff in the end, and he's been on it. And it's still argued about. I think the favoured thing now is they do know when it started. There's been big arguments for a long time. When did oxygen come into the Earth's environment and atmosphere? And that's been pushed back and back and back now. And it came in, if not right at the beginning, but very soon after that. Because for a long time, the banded iron formations, which occur all over, they seem to be stuck around the 1.8, 1.9 billion years. And that was thought to be the kind of reservoir of gathering all the oxygen. And these have been found that there's some good evidence in these Greenland materials that, way back, the banded iron formation. So I think that we do know now when it happened, but I think it's always healthy that when we're working in origins of life or whatever like this, when you're working at the frontiers, I mean, it can be any subject. I think people want to ask and should ask more detailed questions and not just accept it. When it's the oldest or the biggest or the whatever, and we're still in the asking more questions about it.
Any other questions?
I have a question, Jim. I was interested in your biomarkers that you showed us.
Yeah.
Is that a one-way process, or will the biomarker flick back if the sample cools down before you've had a chance to look at it?
No, it doesn't.
It only goes one way?
Yeah, yes. The ones that we know about, and I think that's quite correct, because they reach an energy phase, and once they've gone over the top of the energy phase, then they don't go back.
Right.
Yeah. But you may have another biomarker which is similar, which it won't go back, but it'll do its switching a little bit further up, or when it's cooler. So those arrows that I'm pointing down, where 45, 65, 85. That's why some of them can be used at different stages, but they don't go from 85 back to 45.
Right.
No.
Thank you. No more questions from our online participants.
Okay. We must have covered everything then. Thank you very much. On behalf of everyone, thank you very much for covering such a wide range of topics. I think we've covered all the sciences tonight, so thank you very much.
And thank you to my projectionist.